I already ended that. That's no worries, no worries. You asked for that, got it. Perfect. So I was trained as a biomedical engineer in Eindhoven, where during my master's I ended up in Maastricht. I don't know, you probably don't know, but the biomedical engineering faculty in Eindhoven, University of Eindhoven, was actually set up with the Maastricht University, which was a health sciences university. So when I was studying there in the early days of that faculty, I really had teachers from Maastricht learning about the human body, and teachers from Eindhoven learning about engineering and the basics of that. A little bit about Karen. Of course, we are very proud of Karen. We are cardiovascular people, including myself. Am I still an engineer? I don't know, judge yourself, but I've been deformed quite a bit due to my environment. That means that I can pretty much talk like a cardiologist or a physiologist nowadays. But I can also adapt, of course, because by training I'm an engineer. But I have to, because if you want to survive in an institute like this, it's not easy as an engineer. So you really have to adapt to your environment. And that starts with a language and also presenting. If I present our model with the equations and everything, then they send me home, they don't listen. So this is some key figures. I think this is one of the largest cardiovascular research institutes in Europe, maybe even the largest. And our research is really organized in blood division, vessels, and heart. I know in Amsterdam here there's quite some thrombosis simulation research ongoing, but I'm a heart person. So we have a model of the heart and the cardiovascular system that I will show about. And that is, well, it's hard to see. You can better use the mouse. This heart division here has two focus areas, which is structural heart failure and complex arrhythmia. And as a modeler, I really like that because I consider myself to be in between, because we can link the pump function of the heart to its electrical activation or function. And that is really, I would say, a relatively unexplored area in experimental work, because it's hard and complex. So to start with the acknowledgements, this is my team, and I'm showing that because it really is a mix of people with an MD. Here I have 3 people with an MD training. They're more on the application side and taking care of data, and the rest are all engineers or mathematicians trained abroad or in the Netherlands. And what I am working with really on a daily basis is people from the cardiology department, people from biomedical engineering, where I'm myself, and the physiology department. So that also really influences our work. And when I talk to clinicians, which I usually do, actually you as an audience are, but I don't present very often to non-clinicians, I have to say. I always tell them, you know, modeling is also your job, because a cardiologist having a patient needs to know what's wrong. And if you know what's wrong, you can treat it. And what typically is done, of course, is you can do all kinds of diagnostics, all kinds of non-invasive diagnostics. So here in this case, you can see an echo with different measures. You can look at blood flow. You can look at how the heart deforms. You can measure that non-invasively. Or you can take an ECG, giving some impression on the electrical activity of the heart. And what they do up here in their computer, they're actually integrating and sort of modeling in a different way than most of us do. And then you try to interpret that and derive properties intrinsic to that pump, the heart, or the system from these measurements. And just to get grip on what's wrong with the patient and where can I intervene and where not. So you could see that as causes and effects, and I'm sure you're familiar with that. You can describe that in a mathematical way. And obviously we use a lot of basic physics law, Ohm's law, Laplace's law, Bernoulli, and Fick. Those are principles that are at the fundament of what we do. But the heart and the whole system is very complex. It's really a non-linear dynamic system. And to describe that in an acceptable amount of time, I would say, is not easy and is not a trivial thing. And in Maastricht, well, you said I'm the lead developer of CircuitAdapt. CircuitAdapt is the name of our model here. But actually, I'm the lead applier, let's say, because my professor, Theo Arts, who is already retired for quite a while, for 10 years now, he really developed the model in the basis, and we're expanding it, of course. That's a continuous process and mostly applying it, but all credits go to him and his knowledge on that side. It's not so easy to link some biophysical properties like a membrane potential of a muscle cell, which our heart is a muscle. So it's getting electrically stimulated, it has a membrane potential, and how that then leads to a calcium release. Calcium is necessary for contraction. How that then leads to force development, and how the force development leads to a pressure buildup in the pump or a decrease, which leads to flow of blood. That is basically the chain of events that we try to capture with mathematical equations based on the knowledge we have. So it's really a knowledge-driven model I'm talking about, not a data-driven model. And so in essence, okay, so one major point here is that it's multi-scale, so that we really mimic the actions at protein and cell level and connect that in a hard way, mathematically hard coupling all the way to blood flow through the system. But for today, we're not going to dive into the subcellular processes, which we do also model. But depending on the research question you address, you do not necessarily need these complex subcellular processes. So for today, we stick to the pump and system level. So in essence, CircuitAdapt is a real-time simulation tool. It's freely available, so you can go to the website, download the source code if you like, and I'm going to show the simulator as well later on. And it actually started as a teaching tool. So it was by Theo Arts, it was developed as part of a course, an engineering course. So engineering of the heart and the blood vessels. And he gave students, that was in the 90s already, he gave them an assignment to make relatively simple models describing the function of a blood vessel or a valve, a heart valve, or the myocardium. And then in the end, he himself, of course, optimized these little modules, put the thing together, basically, and ended up with a closed loop system. Nowadays, we really use it for collaborations with device companies. I don't know does anybody know Medtronic? Yeah. Medtronic is a company making pacemakers, for example, insulin pumps, these kind of things, with quite some cardiovascular devices. And they, on a regular basis, reach out to us to test hypotheses, let's say, and thereby they use that in some way or the other. Usually I give them the results and then it stops. They don't tell what they're gonna do with it, but
But actually, they are one of those companies who are a bit more easygoing with these connections.
Yeah. That's. I also profit from his history, because actually Medtronic has the only research lab outside Minneapolis. That's where the headquarters are. Is actually in Maastricht, across the street of the hospital.
That's convenient.
Very convenient. So there's actually research being done there. And the reason for that is that Maastricht back in the days was, and still is, clinically really big in EP, electrophysiology. And well, Medtronic started as a pacemaker company. It's the founder of Medtronic, Earl Buchan, who invented the pacemaker in his garage, by the way. And so then also that research institute is called the Buchan Research Institute. So we collaborate with Medtronic quite intensely, and with the model like this, I'm just happy to have them close by. But also we use it as an educational tool, and some basic thoughts behind it, at least that we find important. I'm not saying that they are important, but that's why the reason, we are in a health institute. It's a knowledge-based model, as I said. It should be physiologically accurate, obviously, so there's quite some validation going on, but we want to maintain a certain low water characteristic, so it should not be too complex. And the reason for that is calculation speed. It's really fast, and when you start to, if you want to personalize models, I don't know if anyone is doing that, but then you get into the computational cost problem. So the more difficult equations you want to solve, the more cost. I probably.
Is it okay if you ask questions now?
Of course, go ahead.
I was wondering, because you also talked about the multi-scale aspect of this model of all these modules. I could imagine that the clinically relevant time scale is different for all these scales.
Yeah.
When you're talking about this fast or that fast, is that for the whole thing, or you specifically talk about some modules?
So I, purposely, on the previous slide, said today we're going to talk about this levels of detail, because if you add the EP model to it, ions that move across ion channels and so on, you need a much higher time step or a time resolution than mechanics. Yeah. So we are not working on personalizing the model, including the EP, because that's, it's possible. There are groups in the world doing it. It's not really done by us, and we also don't have the ambition to do it. We really want to focus on mechanics and hemodynamics. Yeah.
And the coupling itself, you mentioned it's hard coupling. What does that mean?
That means that, so we have now, and this is unpublished, so we are about to submit a new model that describes the coupling. I mean, a heart cell, a myocardial cell, is being electrically stimulated, then it releases calcium, and that calcium binds to a protein called troponin, and then you get interaction of proteins in the cell that generate force. But if you stretch that cell, it also leads to calcium release from those proteins. So there is a two-way interaction between electrics and mechanics, but also if you change mechanics suddenly, you change electrics. I don't know if that's a popular, not so nice, but a popular example. You may know these YouTube movies where ice hockeyers get a puck on their chest and they drop dead. They have ventricular fibrillation. That is an example of where, when there's a sudden impact to the chest at the wrong moment, at the wrong time, and the wrong space, that a mechanical stimulus also affects electrophysiology and you get into ventricular fibrillation. That is what I call hard coupling. We can also say we have a model that produces a calcium transient, And that calcium transient is the output of that EP model. And then we feed that into the mechanics model to steer it. Then there is no interaction back from the mechanics to the calcium. And that is what we call soft coupling.
Right. Thanks.
Yeah, so, but I want to go in the end of this talk, I want to show you some personalization cases of real patients that we try to mimic in the computer. And that's why we start at force, and we have a phenomenological model that describes the generation of force in a myocyte, which is relatively low order, and thereby it's very fast. Okay, so and what are we talking about that then? In that configuration, we have in MATLAB, but you know MATLAB is very slow. But we started developing everything in MATLAB. Now we have implemented everything in C++. And in April, we're releasing actually a whole platform where people can download the source code, the C++ code. And there's a Python wrapper around it. But it makes it's almost, depends on what the configuration is, it can be 1,000 times faster than real time. So then doing a large scale sensitivity analysis, for example, is possible. Okay, it's modular. So this is what I said. These modules represent, for example, a valve or the function of a blood vessel, having a compliance, being nonlinear in its behavior. The more you pressurize blood vessel, the stiffer it becomes. That sort of knowledge is all in these modules, and if you put that together, you couple this, you end up with a closed loop circulation, as we call it, like our own cardiovascular system. So what is that then? It's a system of ODEs. It's a 0D or 1D configuration. You can discuss about that. We have also a more advanced blood vessel module that actually has a wave propagation description. So then it's 1D, because then the vessel has a length and a forward and a backward traveling wave, like in the real blood vessel. But we can also have a more simple configuration where it's 0D, and then it's faster, and for certain purposes, then it's fine. So, but still, it's a unique model. There's not many models that describe the hemodynamics and mechanics of the heart in this way, in such a complete way. So this is Emeritus Professor Theo Arts. One of the very specific and unique features of this model, and that's why it's called circ-adapt, is that there is a specific feature which we call mechanofeedback. So we tried to build a feedback control into the model, that if you, for example, create hypertension in the model, which is easy, you just increase a resistance, then the heart has to work harder. If that would happen in one of us, then after a few weeks, our heart starts growing. It starts hypertrophying. It's like you go to the gym and you do exercises every day, then you get bigger muscles. That happens with the heart as well. And that is very hard to model. Because that goes into the subcellular protein turnover and so on. So we created a model, a relatively low order model, where the circular model actually produces itself the load of the tissue, and then having certain reference mechanosignals, it can either grow or shrink or get stiffer or more compliant, or it can actually dilate or get smaller. And by having a mechano-sensing feedback system, we call that, we can actually have a model that predicts the remodeling of the heart given a certain change of its loading condition. And that's what's happening in patients. Or in patients, this remodeling derails. Okay, so I wanted to show you what this then looks like. Let me see. We have I think I shared the screen, isn't it? Yeah. So 10 years ago or 12 years ago, we actually decided to create a shell, a user interface around this code, because I'm in a medical faculty. I'm actually teaching cardiovascular physiology to medicine students. And we came up with the idea to create a shell around it such that medical students can actually change properties of the cardiovascular system and then see immediately how pressures, volumes, blood flow across valves, how it responds to that. And usually medical students learn this from textbooks, but here they can create their own diseases and immediately see how this the pump function basically responds to that. So you see here the pressures in the left heart, for example, and if I can add whatever pressure I like. And then the fun part is that you can, oh, it's on the wrong screen. You can change properties. So for example, if you, it's a bit hard, it's too small, sorry for that. But for example, here we have the 4 valves in the heart. And if you take the mitral valve, which is the valve between the atrium and the left ventricle, you can actually make it leaky. So now it's not leaky, it's closing properly. But if you make a little hole in it, it is a patient with mitral regurgitation, what we call. And if you do that acutely, so I make a hole here. So then you see that this is here, this is how we started. This is the healthy heart with a normal, healthy mitral valve. And if you make a little hole in that valve, then you see many things change. You actually, and then if I teach this, then the students can really step by step dissect what is happening. Because if you change one property of the valve, it's the atrium that feels it and behaves differently. It's the ventricle that feels it, that behaves differently. The output of the ventricle is less, so your pressure drops. And that is what you see here acutely happening. And we have something like flow velocity across valves, which cardiologists do measure on a daily basis with a Doppler measuring Doppler signals in an echocardiographic exam. So they really learn to interpret the integrative function of the heart, which is super complex. Because if you change something in the right heart, in the left heart, everything changes as well. So if you would not understand the interaction and the dynamic interaction of the system, and you see something has changed in the left ventricle, it does not necessarily mean the problem is also in the left ventricle. You see?
Right. Can I add? So I'm just wondering, because when you teach this to medical students, of course, they are not that interested whether it's MATLAB or Python, but to what extent do they believe this? Right?
Oh.
So you show this dashboard to them and then they start to turn the knobs and learn what's happening. They just take it granted that, okay, this really is reality.
It's like, I think they believe this more than textbooks. Because in textbooks, it's just described that this is happening. Now they are doing it themselves and they see it. And
but
you know the
critical
simulation
behind.
Sorry?
They do know there's a simulation behind it.
Yeah,
they just accept that.
Yeah. And the simulation is based on well accepted principles, like, inertia, for example. Nobody doubts about inertia. Question is, how is it implemented, of course? But that for medical students, doesn't care. They are actually being taught to learn patterns, to recognize patterns. This is actually what the teaching leaders in Maastricht really like, because this is not learning patterns. This is learning reasoning. And that is what they miss. Yeah. So the fun part is, I'll get back to you. The fun part is that I made practicals for first and second year medical students. We exposed them also in the third year. So when you studied in Maastricht, I think the average clinician believes in simulations much more than anywhere else. But the thing is, I actually gave exactly the same practicals to cardiologists in training, which are 6 years older. Their level was not better, I can tell you. It's really, really true.
So I wanted to ask, so there's one way to study this, like using these simulations, and so the other way would be, do people just look at case studies to understand these different, like say, there's a hole in the wall, as you said. So would they look at case studies to understand what would happen in different?
Exactly, but that is what comes later on in their training. Because if you have a real world patient case, a patient never has only a little hole in the mitral. This patient also has an infarction, has atrial fibrillation, everything together. But having this tool then to see what do the isolated diseases do, it makes the student much more capable of integrating in the later stage. At least that's what we hope. But yes, I mean, getting back to your question, you see, because we do, we have 380 first year medical students every year, but you see a divide. Some really like it and they love it. Those are more the tech oriented students, and some really, they really don't give a shit. If you would give them a book, they would be happy to. It's really sorting out different ways of thinking. And I think the ones who like it in the first year, I never did the research, but they're most likely to become the more technical specialties, like cardiologists. They are more beta sort of people. Yeah. Anyway, so this is very simple. You can make it very advanced and very complex. You can change tissue parameters even locally in the heart, so you can create a myocardial infarction. You can look at strain and stress in the tissue, which is not displayed now, but we can. So that can be done here. So it offers a lot of opportunities, but in general, students really like it. And in the Netherlands, in Nijmegen, they're also using it. In Utrecht, they're using it. It's freely available. So if you know someone in Amsterdam, just tell them and they're free to use it.
Can I ask a question?
Yeah.
How do you define the reference? Because I'm assuming that is there one ideal heart that you use as a reference?
You can come to our lab, and this is a discussion we have with the whole group. We're trying actually to define something everyone would consider normal. That doesn't exist. That's really difficult. And then also to find the normal ranges and parameterize the model such that on all signals, flow, pressures, volumes, it fits in the normal ranges. That's the science apart, but we're working on that. This year has been sort of obtained manually so far. Okay, but it's acceptable, It's more than acceptable actually, because it's really considered by everyone so far being exposed to this as more than okay. Okay, let's go into one little application, and this is a research application. Well, I don't know how familiar you are with the heart and how it works. But in the heart, we have a fast conducting system that makes sure that every part of the heart, and the heart contains 4 chambers, basically, that all those chambers are electrically stimulated at the right moment and thereby contract at the right moment. So we have the normal heart here. If we once again play this animation where this electrical stimulus starts here and then is conducting in a fast way to activate the left and the right ventricle in a synchronous way. We have a disease, and many heart failure patients have this problem. It's left bundle branch block, where actually the heart, the right ventricle is activated in a fast way, but the high way taking care of the fast activation of the left ventricle is actually damaged. So it has to be activated from cell to cell conduction rather than through the fast conducting system. And that's how you end up with a situation where this part of the heart, this part of the muscle, is actually very delayed in its activation, electrical activation. So we have a treatment for that, and that's one of the treatments, for example, Medtronic came up with. This is cardiac resynchronization therapy. So it's a pacemaker you implant in the chest, and the pacemaker actually senses when the atria are activated by the heart itself, and by the sinus node. And then it says, well, with a certain delay, we're going to pace this right ventricle and the left ventricle to sort of restore the synchrony of the 2 ventricles. The problem, though, is that actually in quite some patients, this does not really lead to an improvement of the patient thing. And with pills, that's not a big problem. They would really, pharmacological companies would be very happy with this success rate. But with devices, it's slightly different. You cannot implant a little bit of CRT. You implant a pacemaker, and if it doesn't work, it's also very risky to take it out. So let's now try in our model, and this looks very complex, but the way to look at it is that this is what the model simulates at pump level, so pressure volume. That pressure volume, you can describe the action of a pump with the pressure volume loop. We have flow across the valves, we have the volume of the chambers, the pressures in the chambers, and it actually simulates what happens at the tissue level, at stress and strain. The myocardial cells generate stress, and that then leads to pressure in the cavities. That works quite well if those 2 walls here, this is the left ventricular free wall, and the septum, that's the muscle in between the right ventricle and the left ventricle, when those walls are activated synchronously. Then you see that stress, strain, so this gives work actually of the tissue, that it's quite homogeneously distributed. Now, if we have left bundle branch block, and these simulations I've done, that's almost 50 years ago, but if we delay the activation of this lateral wall just by 10, 20, 30, 40 milliseconds, which what one can consider as left bundle branch block, then you see at the tissue level, you see, I'm increasing this delay here, the black wall. You see at the tissue level, very different mechanics going on. It's the late activated wall here that performs much more work than the early activated wall. Whereas at the pump level, hardly anything has changed. So this is what has, it's quite complex. If you go back to the synchronous situation and you look at the strain patterns here, shown here, this is what a normal heart should look like, means that it's shortening during the ejection phase and it's lengthening during the filling phase. And all the muscle surrounding that cavity is doing that in a coordinated and uniform way. But if you delay one half of the heart, you see that that behavior is messed up tremendously. You see here, this whole septal pattern is completely different than the late activated pattern. And this has been validated with dog experiments. So in Maastricht, we actually did experiments in large animals. And the reason why it was dogs is that the conduction system of a dog heart is very similar than the human heart. So you cannot create the disease left bundle branch block in a pig, for example. Impossible, because they have a completely different conduction system. So back in the 90s and 2000s, they did a lot of experiments to understand what is going on here. And you see that if you create left bundle branch block, here is the septal behavior, and here as well, versus the lateral wall, the free wall behavior, that's the dashed line here. And that's exactly what our model also shows. So based on simple physiological laws and physics, you can actually describe this mechanics quite well. And well, there are lengthy validation studies of that. So those large animal experiments have been very useful for us to validate the model at the tissue and pump level. Then, after that validation, a group of cardiologists approached us. They're from Utrecht, so your Utrecht UMC. And they said, well, we actually have heart failure patients, 132, with left bundle branch block. But when we measure the strain, so the deformation of the septum at that wall in between the two walls, it looks completely different. We know it should look like this in a healthy control, but we have patients with this pattern. And you may recognize that pattern because that's the one the simulation showed. We have also this pattern, which is in essence very, very different. It stretches all the time, and it shouldn't be like that. It should shorten. And then we have patients that behave like this as well. So they sort of saw three categories. So they asked us, can you use the model to give explanations for this? Well, we had some candidate hypotheses, what changes in those patients. And the first is very easy because there is a delay of the lateral wall. That's what we already showed. Then if we make it desynchronous, the lateral wall, you see that the septum starts behaving completely different, like the way I already showed, but it mimics very much what is shown here in the patient, what is observed in the patient. And this also, to me, shows how complex understanding this is, because I'm actually changing a parameter of a tissue of a different wall. But because I'm changing the delay of the lateral wall, the boundary conditions of the septum completely change, and thereby it starts behaving completely differently. So that's what I mean, where models are really useful in understanding what actually is going on at the tissue level in the heart, and thereby also being better able to interpret diagnostic measures like strain patterns. So how now can we get from this situation with a delay in the lateral wall? We shouldn't touch that because the patient has left bundle branch block. So there is clinical evidence, there is a delay in activation here. So then when we started to weaken the septum, so make it less contractile, you see that that same pattern starts to behave like observed in these patients. And if you would even add failure of the myocardium in the lateral wall, you see that that pattern changes into something really looking like observed in these patients. So this is what we call forward simulations. This is, we do not provide evidence here, but we test hypotheses and show this hypothesis, this underlying disease is more likely than this in this patient. So this really helps to interpret those diagnostic data. What we didn't know in the beginning is that the Utrecht investigators also knew that these patients respond better to CRT than these patients. And if you look at this from a modeling point of view, that makes sense, because the model predicts that those patients have much worse myocardium, failing myocardium. I won't go into detail because even at the subcellular level, we can pretty much explain this nowadays. But this brought us to the idea to actually see if we can use virtual patient populations to create a diagnostic index, a number a cardiologist could measure in vivo with echo to predict what happens when you implant a CRT device. So that's what we did based on some quite extensive sensitivity analysis. We came up with this index that calculates the pre-stretch in the lateral wall and the rebound stretch, as we call it, in the septal wall. It's just a number you can measure with echo, and we did that in a large database, and this is the whole pathophysiological and biophysical reasoning behind it, why this would be a good number to predict CRT outcome.
And what was the order of, so was it like you did a big sensitivity analysis, then these two quantities of interest popped out, and then you went to find some well-educated reasoning why this might be, or was it the other way around, that you knew that, okay, these must be important for the outcome of this pacemaker implantation? So we need to look for indices that are related.
Yeah, it was the latter one, because this study that I just showed you was done already. So we knew already that those are in fact three parameters, that they had a significant effect on the measure we are getting from the clinic. But then we had to be sure that not all other parameters in circuit duct could also influence this. So that's how we did a sensitivity analysis, basically to hopefully conclude that indeed those parameters were the most important.
Right.
So that's the way it went, really, and that's often the case, of course. Okay, what we did, we actually measured this number, this systolic stretch index, in a cohort of patients, and I don't know if you're familiar with Kaplan-Meier curves, but this basically says in time after the CRT, the pacemaker was implanted, how long it takes that patient either get hospitalized or actually die. So the faster it declines, the faster the patients die, basically. So we saw that indeed, with having a very low SSI, you have a poor outcome here. Having a high SSI, you have a very good outcome. And this is as a control measure.
Is that a good outcome? It's just 2 years from the implantation.
Yeah.
And it's already 2 out of 10.
Yeah.
Okay.
So here you see that half of the patients die. Is it good? Maybe I said it wrong. It's not good. It's much better than the others. Huh? But you know, it's a devastating disease. Those patients have heart failure. Huh? So, yeah, well, good point. It's not a good outcome. Certainly not. Looking at death, then you see that same thing holds if you select those ones at baseline, before you implant the device, and you have high SSI, then you are in 2 years quite, you know, there is a dropout of only 10 or 5%. So that's quite good.
Sorry, what's the black line?
The black line is actually, that's a whole story apart, but those are the patients the clinicians at the moment don't doubt about. So they have certain diagnostic criteria. There is no clinically relevant question whether or not the patient should receive a device. We only selected the patients where the clinicians, by guidelines, basically flip a coin. They don't know what's going to happen. So that was, for us, for our diagnostic marker, the most interesting population, because there you can really add on the clinical decision making.
How do we signal that? We signal that somehow?
Yeah, from an ECG. So you can have certain ECG parameters, and an ECG is easily obtained. That makes if you look for certain characteristics in the ECG and the patient has those, then you know almost for sure that this patient is going to respond, and that's the black line. Yeah. Okay. What time is it? Well, it's almost 5 already. So I'm going to skip this part. We're also doing some more advanced pacing simulations. But the last part I really want to show you is this, because now we can also measure the regional strain in the entire left ventricle, and then see, hopefully, whether the model can tell us what's wrong with the tissue and where. And that is creating a digital twin using a knowledge-based model in this case. And here you see, for example, a heart failure patient receiving CRT. Before getting the device, the regional strain patterns, this is the left ventricle, how they look like. And then you see it's a spaghetti, you know, all those lines should actually show one and the same pattern. It will probably do in our hearts in case we're healthy. So this case is definitely a heart failure patient. The question is, how is actually the myocardium doing? And we want to create with our model a simulation that really behaves like the one here. And that's not so difficult, because our model has about 200-300 parameters, so you can tweak it and it looks like what you observe. But you know, the problem is this a reproducible result you get, and is it a unique solution? And that's what we have been working on quite a bit in the last 5 years, and we have sort of a framework. It's a parameter subset reduction framework. So let's say we start with the entire circular model. It counts in this setup about 270 parameters, but you will never be able to estimate those parameters from those 18 strain patterns and some volumes that we entered. So this is the limited clinical information we have. It's volumes and regional strain. And then we set up a Morris screen for sensitivity analysis to do some early model reduction steps. And then we're going into a loop, basically doing a particle swarm optimization. We do that 5 times and look for reproducibility. Well, here, this is a subset of 150 parameters that went in there, and then reproducibility is crap. It's really, really bad. So if you then look for the reproducibility and look more into detail, you know, which are precisely the parameters that are not uniquely estimated based on the information we feed the model with, then we're gonna remove them from the subset, and then we run the loop again until we have a situation, and that's our result. This is unpublished. It's going to be submitted next week, where we want to have a compromise between goodness of fit, how good the simulated patterns mimic the observed patterns, but also the identifiability of the parameters and reproducibility. And that's what you see here in those 2 plots. This is actually parameter reproducibility. And you see we started with 270 parameters. And in the end, we managed only to get 75 parameters from the model to reliably being estimated from the strain patterns we have. It sounds like many, but it's not actually, because we have 18 time strain patterns. So it basically means that at a regional level, we can only estimate 3 intrinsic properties of the muscle from a single strain pattern. So that's what you see here. And then you see also that the goodness of fit is very acceptable. If it's above 2 here, then it's unacceptable. So here we have a good reproducibility with a good interclass correlation, and we have a good fit still. So how then, well, this is what the parameters are. I'm going to show you the results. So how does this look like? So if we start with a homogeneous heart, it should look like this. This is an example patient where we did this analysis on, and this is a patient with myocardial infarction. So you don't have blood supply to a part of the heart. And then the day after, they make an echo. And then you see that some parts do not shorten here, this purple one and this purple one, the green one here. And then we asked the model to reproduce this with having those 75 parameters as degrees of freedom. And then what we get out as a contractile dysfunction, so this is an independent output of the model. You see that in this part of the heart, there's really compromised contractile function. That's the red area. So if the same patient comes back 6 months later, the echo is made again, and then you see a different pattern. You see some segments here that are really bad. They're being stretched, do not shorten at all. And if we then apply the model to that, the model nicely indicates that those segments are basically scar tissue. They do not contract, and they're much stiffer than the rest of the heart. Well, and that is also here confirmed with MRI. So this is an independent validation set. With MRI, you can actually localize where myocardial infarction has happened. And that's here. And that qualitatively matches quite well with our simulations. So this is being applied now to quite a data set, and we're about to publish that. So in summary, well, I'm not going to read all these conclusions, but I tried to give you some examples of how we use the model in a forward way to just improve our mechanistic understanding of cardiovascular diseases. And also now in a, but that's early work still, in an inverse way, and trying to solve the inverse problem, loading the model with non-invasive imaging data to actually get more insight into the disease the individual patient has. Okay, so much more we can do. As I said, the model actually is now about 25 years old. So there is quite some development in there. There's a coronary circulation in there. You name it, we can do it. That's certainly not everything. So it has definitely quite some drawbacks. Also, the geometric, simplified geometry for certain research questions is a drawback, and you really need a finite element model for that. That's also actually implemented with another group in Europe, where we have the circulation model around a 4-chamber finite element model that works quite well too. Final thing, this is a conflict of interest. I'm a deputy editor of this new journal. So if you're in the cardiac field and you're doing digital health stuff or AI or anything, then please consider this journal. It's now 2 years old. It's PubMed indexed. For me, that's important because I'm in a health sciences institute, of course. The editorial deputy editors and the editor-in-chief are Dutch. So take that as an advantage, I would say. Okay, questions. Thank you.
Right. Well, I have a bunch, but let's, in the meantime, ask also the online audience that if you have questions, then please raise hands and I will go ahead. But if that happens, so yeah, please keep an eye on that, how that develops. I have a bunch, some I will keep for the snacks afterwards.
Mhm.
But I was really wondering when you were talking about digital twin version and also personalizing it. So it's been a model in development for 25 years. It's a very long time.
Mhm.
Is this now used as some sort of a certified model, or is it being
Yeah.
pushed towards that? Or what
Very
is it?
good question. So the guy who actually started with the whole personalization pipeline, that's Nick van Osta. He did his PhD with me. He worked now for 5 years in my lab. So he's a postdoc now, and he's exactly working on this. So we actually broke down the entire model because we were 3 years ago still using MATLAB. It has been thrown in the bin basically. Wow, that's a bit exaggerated, but he completely re-implemented the model in C++. And now we have a whole platform where we do very clear verification and validation steps. So we have benchmark toolboxes developed now, and he's working on that full time. You probably know that's a massive job.
Yeah, I've heard about it. Yeah.
But we are far with it. And as I said, in April, he really wants to launch this verified and validated toolbox, which is basically a platform. It also has, for example, a more screening tool that we can apply to circuit depth and so on. Yeah.
And do you follow some sort of because the guidelines in how do you certify such a model is still a little bit dynamic,
Yeah,
flexible,
yeah,
and so
yeah.
the gray zone. And in the States, they are a little bit further ahead with this VNV40.
Yeah, it's the VNV40 guideline that we apply.
Yeah.
But as you say, at certain points, you know, there is not one way of doing it. It really depends on your model that you're working on as well. But as much as we can, we follow the VNV40.
Mm-hmm.
And there is a good reason for that also, is because now we are in the process of doing a specific study in pacing. I can't tell much more about it, but it's so new. We're actually using model simulations to define a clinical trial design. And if you do that with a non-verified, non-validated model, you can run into problems. Just, you know, you need a certain level of verification.
That's super interesting. So you're not talking about setting up an in silico trial, not even a hybrid one, but using in silico tools to design a real.
Yeah.
Okay.
You know, well, for example, show which inclusion and exclusion criteria you should apply. That's what I mean. So it's not that we push a button and then there is a trial protocol. No, of course not. The trial protocol also involves prior knowledge and so on. But if you make very clear and quite important choices based on inclusion, exclusion criteria, based on simulations only, that's quite a new field. And then you can run into problems if you didn't do any verification of your model for good reasons.
Right.
Yeah.
Okay. So let me see.
I have a question.
Please.
Yeah.
Yeah.
So I was, well, first of all, yeah, very interesting work. I'm not in the field of
The cardiac. Yeah.
just more on the simulation. Really interested there. So I was, I guess, first question is about the model. So you have these very different parts that at the different time scales and physical and spatial scales potentially could. And you always talk about one model.
Yeah.
But is it more like different pieces And then how
Yeah,
do you put
you
them
know,
together? Do you have a platform to run them simultaneously?
it's great that you asked that, because this is an evolving discussion topic. You know, I've been using this model for 15 years now or more. And at that time, it was just simply a MATLAB package of, you know, like we were plumbing basically and putting, and then we called it the circuit app model. But now we actually broke it down and re-implemented it to be really modular in terms of programming language definition. Now it's not a model anymore. It's like a library almost. It's a platform. So my postdoc Nick, he is actually planning to publish this as a platform with, you know, making everything open source. And we have lengthy discussions how to call it. A platform, a pipeline, not really. I mean, a pipeline is more what I showed about model reduction. That is a pipeline where you have a certain recipe of how to follow this.
Framework is very
framework.
old school. Framework is very old school. Like,
Yeah, so if you have suggestions, please send them. We don't know, but we do know that being stubborn and keeping it calling the circuit app model is probably not what it nowadays entails. It's more than that, but that's history.
And no, I think that's I guess my question had to also in relation to like how for the climate models, which is I guess for you also have this idea that you have not just that they are modular in themselves, but you can replace pieces of one model for another, and then when you run these simulations and you try to make the policy, you actually,
as long as input and output are
similar,
similar,
within the same, then you can replace them for different models.
Works the same here.
Okay, and then you use a platform to integrate these models, or
That's what he wants to publish. And for example, we have a blood vessel. And that blood vessel you can describe in several levels of complexity. And we have two blood vessel modules that you can either plug and play, depending on whether you need the model complexity to address a certain research question. So we adapt now the complexity of the model to the research question. And
So if you ever need a cellular level model, then let us know. We might have that. And just a small thing, when you mentioned that it was coupled to a more finite element heart model, was that the Barcelona group with Alia? Graz, okay.
Gernot Planck.
Okay, I don't know that.
No.
But okay.
Yeah.
All right.
Ed Wigmund and Gernot Planck. So that's Bordeaux and Graz, University of Graz.
Right. I will have questions on that offline.
I have two more questions, or maybe offline, or
I think we can open
time.
some beers in the meantime, right? That would be more fun to do
Good
that
idea.
way.
I'll stop the recording.